All right, welcome to Enoch's videos. Today I want to talk about the models. We normally use models in biology, and today specifically we're going to be focusing on the models of the cell membrane and what are our main objectives today, or what are you supposed to learn today. One of the things that you must learn today, you must know what are scientific models, why are models used in science, what are some of the types of scientific models? Why do models have limitations? And why do models change over time? That will take us about 15 minutes. And also, you must be in position to give your responses on those main objectives I've just highlighted. Our main objectives, I'll share them with you. I said you need to know what are scientific models, why are models used in science, what are some of the types of scientific models, why do models have limitations, and why do models change over time? Right. So we need to know what are scientific models. So models are conceptual representation or representations used to explain and also predict phenomena. So in explaining, what are they explaining? They're trying to explain what has been observed, or they're explaining observations. Also, they are used to predict future observations. That's why we use models. And mind you, models are supposed to be realistic. So models are explaining processes or complex processes, complex structures that cannot be observed nor experimented. So there are processes that cannot experiment, and in such case we use a model. Or there are some structures that are too complex to understand, and we still use a model to explain them. That's why we use what? That's why we use a model. Okay. I hope now you know why we must use a model. So why do we use models in science? Why do we have to use models? So models are very important. Why? They can simplify complex structures and processes with ease. This can be explained with ease. Then also, we find that models can change the scale of objects that are too big or too small to see at all. Imagine, look at the Earth, but we have a globe that is used in several classes to represent the big Earth. There is DNA. We have the models. We have the heart. We have the model. It will be too complex to study the real structure of the heart in its reality, but we have a model. Uh, so models change the scale of babies that are too big or too small to see them at all. Models can be used to change the rate of the phenomena, can either slow it or speed it up. For example, if it is digestion process, can decide to speed it up and see what takes place within a short period of time, or slow it down to critically analyze the phenomena or the process. I want to tell you that models can help us communicate by simplifying difficult, complex concepts and making them more tangible. Something that was in your mind, that was in a picture, that was something like imaginary, is brought to real life. It is tangible. I want to tell you that science use models, e.g., computer models, to make and test predictions and to understand processes that are not visible or easily observed. Okay, hope we are on the same page. We have several types of scientific models. We have the mathematical models. These use mathematical language to describe the behavior of a system. We have computer models. These attempt to simulate the behavior of a particular system. Then we have physical models. These ones can be touched and held. We have pictures or diagrams developed in an effort to represent something that is still a model. And lastly, we have analogies. That is a comparison between two things, typically on the basis of their structure and for the purpose of explanation or clarification. Okay, so that's giving us a scientific understanding, and we need to always be in position to have the theory in mind in order to come up with a model or a simulation so that we fully understand the scientific structures and processes. This is a caution. We need to know that models have limitations. They may not be in position to explain details of a structure nor details of a given process, which you're calling a phenomena. We need to know that models are not copies of the reality, but they are just representing, or they are just similar to what they represent, but they are not the same as what they represent. We need to also recall that a good model must be able to represent as many characteristics of the reality as possible, but also be as simple as possible. What are some of the limitations of models? One, we need to know that models normally come under scrutiny and also change over time. So they are not static, they must be dynamic, and many times they're under scrutiny. Models can also be as accurate as the current state of the scientific knowledge. So as the scientific knowledge changes, definitely even the models must be changed. Then, models tested against experiments or data from observation may prove inadequate, and in that case, they may need to be modified or replaced by new models. There are some historical models of the same membrane that you're going to look at with specific characters and specific years and what they discovered. Look at what we have on the screen. The first discovery was in 1899. That was by Overton, and he discovered that membranes are made up of lipids. And what did he do? We shall have to discover what he did. We have Gotter and Grendel, that is 1925. This guy, or these two guys, found out that membranes, although they are made up of lipids, but the lipids form a bilayer made of phospholipids with heads which are hydrophilic and tails which are hydrophobic. That's what they discovered. Also, we have Davison and Daniel, that is 1935. For him, he found out, that is Davison and Danieli, they found out that although the membrane is having lipids, which are in a bilayer, but on their surface, there are protein molecules attached to each side of the lipid. That was 1935. Then when we come to 1972, when we find Singer and Nicholson, these guys found out, yes, the membrane is fluid bilayer of lipids with embedded proteins. You get it? So that's what was discovered, and that's what we are going to look at in today's lesson. All right. Okay, unless one has a question. If no question, I'll